Hello, so let's talk about gene editing. And it's exactly as it sounds, like making changes to the DNA sequence. And so we're going to talk about ways you can do it and why it's important. So go back in time to when we talked about the central dogma, and the sequence of nucleotides in the DNA are going to be used as a template to create that mRNA molecule. So this mRNA is going to be complementary to the DNA, and then that mRNA is going to be translated into a protein. Well, it's that sequence of the DNA that dictates what the polypeptide amino acid sequence will be, right? So what happens when it's altered? So think of a single nucleotide disease, sickle cell anemia. There's one change. So in the, let's say, normal DNA sequence, there's a T to A base pair. Well, in people who are affected with sickle cell, they don't have that one. Okay, so there's this single base pair change that causes an A to T. And so under quote-unquote normal conditions, it codes for glutamic acid, whereas in sickle cell, it codes for valine. Okay, so a change in that DNA sequence causes a change in that mRNA sequence, which causes a change in that amino acid sequence. And that single base pair change then changes the structure of the protein, and in this case, hemoglobin. So we go from the disc-shaped normal red blood cells to these sickle-shaped red blood cells, okay? And then all of the phenotype that follows. Well, now we know that there are different ways that these mutations can be introduced into a cell. So some are spontaneous. Something that happens within the cell causes these mutations to happen. Now this can be like byproducts of normal cellular pathways that increases things like reactive oxygen species, which can then go on to mutate your DNA. It can also be induced, whereas it's something in the environment that's acting on the cell from the outside that's damaging the DNA, like a UV ray, which can cause those thymine dimers. Now, fast forward from what we were discovering about how they were introduced on their own, now we know that we can introduce them. We as in scientists and researchers. So through a technology called gene editing, we can go in and put specific mutations where we want them to be. Now, when it was the induced and spontaneous, most of those were random, right? We didn't know what or where it would occur. But with the gene editing technology today, we can specify where we introduce these mutations. Now, think back to when we talked about Beetle and Tatum, because that was something similar that they did to knock out proteins along the way to figure out what was going on in neurospore. Yes, neurospore. So with this site-directed mutagenesis, we are going to make specific DNA mutations. So if you take, for instance, the C at the 7th location, and we're going to go in and say, we don't want this to be a C, we want it to be a G. Now why, right? That begs the question, why would we want to do this? Well, the reason we want to do it is so that we can figure out the effect it has on the organism. So if you go back to the central dogma, if we change this in the DNA, what does that look like then in the protein? So we can figure out how it affects gene expression. We can figure out how it affects the protein function. And ultimately, what does it look like to the organism? So what kind of overall phenotype is formed? So how do you do this? So with site-directed mutagenesis, it requires that we denature our DNA so that we have single strands, and we only need a single strand for this to occur. So we use this as a template strand, and we're going to have this oligonucleotide primer that contains the mismatch that we want to introduce. And it's going to have an area that's complementary to the vector, which has our template DNA. And so we're going to allow those to come together and then use that primer to go ahead and fill in the rest of that sequence, right, to make it double-stranded. And in doing so, we're trying to introduce this error that we have, right? And so we're going to go through the steps of PCR, letting it anneal and synthesize and all of those things. Then we take this vector and we have to introduce it into a bacteria cell. Okay, so this will allow us to see what's going on in that bacteria cell. Or do we want to make more of these for whatever reason? We still have to be able to clone it inside of a bacteria or another cell that would accept it. Okay. So we can do this then to introduce it into plants or animals, depending on what kind of mutation we're creating. That's site-directed mutagenesis for gene editing. So the other part of this is the CRISPR-Cas9. So with the CRISPR-Cas technology, we've talked about this and how it provides this immune system for bacteria against bacteriophages. Well, we also briefly alluded to the fact that researchers have been able to use this to edit genes in living cells. So one difference here is, whereas when we made that primary vector, we introduced that into bacteria before we introduce it into animals or plants. Here we can go directly into a living cell and start to make the changes that we want to see. And now it's important to note that this technology relies heavily on the DNA repair mechanisms. And depending on which repair mechanism occurs, we'll either introduce a deletion, which could fix something like a frame shift, or we can edit the gene, which has been useful in fixing, if you will, single base pair diseases. Okay, so with CRISPR-Cas, you have this tracer DNA, which you link it to the CRISPR RNA. I'm sorry, you have tracer RNA, which you link to the CRISPR RNA. Now, in bacteria, these two things are separate. In the technology that people have went on to create, you link these two things together. Okay, and so this is going to be complementary. This spacer is complementary to the target gene. So it comes along. And once it gets there, my apologies, Cas9 causes this cleavage. Okay, so the Cas proteins are endonucleases, and they're able to introduce double-stranded breaks in the DNA. Once you introduce that double-stranded break into the DNA, one of two things can happen. So on one side, you have non-homologous end joining, where we don't have a homologous chromosome nearby. So we're just going to try to fix this as best we can. And this usually results in inserting a small deletion. Okay, but if it's homologous recombination, now we have a template to go from. And in doing so, we're able to then introduce a change. We're able to edit this gene to create some kind of point mutation, or use that point mutation we're introducing to fix one that already exists. Okay, so CRISPR-Cas9 relies on these non-coding RNA sequencing. Another thing scientists have come up with is using a dead Cas9. And so now in this case, that endonuclease is inactivated, meaning it can't introduce a double-stranded break. So then you might ask, well, what is it used for then, right? Well, you link that Cas9 to a transcriptional activator protein. And in doing so, you don't alter the gene sequence, but you alter gene expression. So this can then be used to increase gene expression, to increase the levels of transcription. Okay, so you don't change the sequence, but you do alter gene expression. Now, those are the main types of gene editing that we'll talk about. So either site-directed mutagenesis, which require processes similar to PCR and require bacteria vector, and then the CRISPR-Cas technology, which can go directly into living organisms. And it has been used on embryos, on mice, on human cell lines, and even roundworms in plants. Okay, so this is actively being used, and trials have been done to see if we can correct disorders like Duchenne's muscular dystrophy. Okay, so CRISPR-Cas9 is definitely a wave of the future, and it should be exciting to see what we can do with it.